Good morning if you are hearing me in the morning, and good evening if you are hearing me in the evening. Today we are going to GIT module lectures. We will start our lecture today with gastrointestinal tract. We are going to talk about the tube, and we are going to talk especially about the esophagus and the stomach. Let's talk about the digestive system. Digestive system is formed of oral cavity. We are going to talk about the oral cavity, tube, which is tubular digestive tract and associated digestive system. And regarding the oral cavity, you are going to take it with musculoskeletal 3 module in the third year. Let's see the gastrointestinal system formed of what? Formed of the oral cavity, which open into the pharynx, go to the esophagus. The esophagus is a tube which becomes dilated to form the stomach. This stomach, after mixing and digesting the food, will pass to the intestine, which part some will be small intestine, the other will be large. Large intestine will end in the rectum, which open to the outside by the anus. For digestion of the food, we have digestive gland, which are salivary gland, which will be studied in the musculoskeletal module. In this module, we will study liver as a digestive gland and the pancreas, exocrine part, which is responsible for digestion. By the end of this lecture, you will be able to describe the histological structure of the different layers of GIT tubes. You will be able to correlate the structure to function of the esophagus. You are going to be able to identify different parts of the stomach and discuss the histological structure of the stomach, including fundic and pyloric gland, and correlate structure to function. Okay. Now we will talk about the digestive tube. What is different part of the digestive tube? Different part of digestive tube will include esophagus, will include stomach, small intestine, and large intestine. As you know, small intestine is divided into three main parts: duodenum, jejunum, and ileum. General structure of this tube. This tube is cylindrical in shape. As you can see, it is formed of lumen and wall. So our wall of the tube will be formed of 4 layers. Okay, and this is special for GIT. Any other tube or vessel, you will find that it is formed of 3 layers. Only GIT gastrointestinal tube will be formed of 4 layers. From inside to outside, you will find that there is epithelium with beneath connective tissue. This layer will be called the mucosa. After that, there is another layer which is connective tissue, and beneath the epithelial mucosa, it will be submucosa. From your previous study, you will find that there is a muscle layer responsible for peristaltic movement. This will be muscularis externa layer. And finally, we are going to have a connective tissue or serous sac. If it is serous sacs, it will be called serosa, or either it is a connective tissue, so it will be called adventitia. So, what is the layer of the gastrointestinal tube? It will be the mucosa, submucosa, muscularis externa, and finally serosa or adventitia. From inside here, this is a tube. It has a lumen. The wall is formed of 4 layers. From inside, we are going to have mucosa. After mucosa, yes, excellent, it is submucosa. And after that, there is a muscle layer, which is muscularis externa. And finally, if it is serous sac like mesentery, it will be serosa, or just connective tissue, so it will be adventitia. Let's talk about mucosa. Mucosa is the epithelium beneath which there is a loose connective tissue. Our epithelium on the gastrointestinal tract will differ according to the function, such that it may be protective in function, like the esophagus. It may be secretory in function like that of the stomach, or absorptive like that of intestine, either small or large intestine. So epithelium, according to the type, will differ according to the site of the tract and according to the function. From it will be maybe protective, maybe secretory or absorptive. Any epithelium, as it is avascular, will be lying over a close connective tissue. This connective tissue will be called lamina propria. After that, there will be a layer of a smooth muscle fiber. Its function is to move the epithelium in order to allow it to extend or take away from the food. This layer is the muscle which moves the mucosa, so it is called muscularis mucosa. It is a very thin layer. It is a layer of a smooth muscle fiber arranged as inner circular and outer longitudinal. What is the function of muscularis mucosa? Is it moving the tube? No, from its name mucosa, so it will move the mucosa. Okay, so what are the layers of the mucosa? There are three layers: the epithelium, lamina propria, muscularis mucosa. What after mucosa? It is submucosa. It is the remaining connective tissue which became divided by the muscularis mucosa layers. This submucosa, it is a loose connective tissue containing blood vessel, nerve, and lymphatic, and it has an important nerve plexus. This nerve plexus is called Meissner plexus. Okay, so the second layer is submucosa. Submucosa is formed of dense regular connective tissue containing blood vessel, lymphatic, and nerve supply. Okay, and there is an important nerve plexus which is called Meissner plexus. Let's go to the which will be muscularis externa. Muscularis externa will be formed of muscle layers, which is responsible for the peristaltic movement of the gastrointestinal tract. So it is more layers than muscularis mucosa. It will be arranged as inner circular and outer longitudinal smooth muscle fiber with connective tissue in between. Also here we have a nerve plexus, myenteric nerve plexus, important for the peristaltic movement. This plexus is called Erbach plexus. Okay, so muscularis externa is inner circular and outer longitudinal. Only no, there is an important myenteric nerve plexus which is called Erbach plexus. And finally, we have serosa or adventitia. As you know from anatomy, there is some part of the gastrointestinal tube covered by peritoneum or some part not covered by peritoneum. So if it is covered by peritoneum is formed of connective tissue covered by simple squamous epithelium. This layer will be called serosa. If it is not covered by peritoneum, it will be called adventitia. So layers of gastrointestinal tube, it is mucosa formed of epithelium, sorry. We have four layers of the gastrointestinal tube. We have the mucosa, which is formed of epithelium lying over lamina propria and separated from submucosa by muscularis mucosa. Then we have submucosa, which is loose areolar connective tissue containing Meissner plexus. Then there is muscularis externa, which is formed of inner circular and outer longitudinal smooth muscle fiber. In between, there is a myenteric plexus, which is called Erbach plexus, and the outer coverage, which is maybe serosa or adventitia according to the peritoneal covering of the gastrointestinal tube. Let's apply this scheme over our first part of the tube, which is the esophagus. The esophagus is a tube extending from the pharynx to the stomach. What is the function of esophagus? It is to conduct the swallowed food, to conduct the food you eat to go into the stomach. Can you say what are the layers of the esophagus? Yes, as we mentioned before, any part of the gastrointestinal tube will be formed of two parts. The first one will be mucosa, and you know that mucosa is formed of three parts. The first one is epithelium. Excellent, very good. The second is lamina propria, and finally is muscularis mucosa. What is important to know about epithelium? What is the function of epithelium? Is it digestion, secretion, or absorption? The function here is to protect. It's protection. So what is the type of epithelium responsible for protection? It is a stratified squamous non-keratinized epithelium. Okay, beneath which there is a lamina propria. What is lamina propria? It is loose areolar connective tissue contain nerve, lymphatic, and blood vessel. Here you have to memorize that. In the lamina propria we have mucus acini. Why we have mucus acini here? In order to make the esophagus epithelium wet. Where we can find this mucus acini? We are going to classify our esophagus into three parts. The upper part, which is related to the pharynx, the lower part, which is related to the esophagus, and the middle one. In the upper part and lower one, we will find this mucus acini at both upper end and lower end. This mucus acini will be called cardiac gland. So mucosa here, you have to memorize that the type of epithelium. It is for protection. So it is stratified squamous non-keratinized. As regarding the lamina propria, there is a cardiac gland. What is cardiac gland? It is mucus acini in the upper and lower half third of the esophagus. What about the third layer of the mucosa? It is muscularis mucosa. As usual, it is formed of inner circular and outer longitudinal smooth to make folds in the esophagus. The second layer of the gastrointestinal tube, it is the submucosa by its content. Submucosa is loose areolar connective tissue, dense connective tissue, which contains, it is a dense connective tissue which contains blood and lymphatic vessel. It also contains mesenteric plexus, which is called mesenteric plexus. Here we must memorize that there is an important gland in the submucosa. It is mucus acini also, and it is called esophageal gland. It is present on the whole length of the esophagus. So here we have submucosa. Submucosa here is a dense connective tissue containing vessels, lymphatics, and mesenteric plexus. This is a general scheme we know. What is characteristic for submucosa? It is the presence of mucus acini, which is called esophageal gland. Okay, now we are going to talk about the third layer, which is called muscularis externa. And here, in order to talk about the muscularis externa, we have to divide our esophagus into three parts as we divide before. The upper third, middle third, and lower third, which is nearer to the esophagus. Okay, smooth muscle fibers of the muscularis externa will be formed of inner circular and outer longitudinal smooth muscle fiber with Erbach plexus. This is what we know. But as regarding this division into upper third, middle third, and lower third, we have to mention that in the upper third, muscle fiber will be skeletal. It is voluntary. You are going to swallow food. It is a voluntary process. So the upper third contain skeletal muscle. The middle third will be mixed between skeletal and smooth muscle fiber. And finally, the lower third will be purely smooth muscle fiber. So to talk about the muscularis externa, esophagus is divided into three parts. Generally, muscularis externa will be formed of inner circular and outer longitudinal with Erbach plexus. This will be very definitely in the lower third. What about the first third and middle third? First third will be formed of a skeletal muscle in the upper part. Middle part will be formed of mixed, while lower part will be purely smooth muscle fiber. Okay? So middle third will be combined between skeletal and smooth. What about the upper one? The upper one will be purely skeletal muscle fiber. The lower one will be purely smooth muscle fiber, and the lower third became completely involuntary muscle fiber. Okay, here can we see that this is a muscle layers. Can here see that this is inner layers, which is inner circular smooth muscle fiber. Outer, you can see here that it is rounded because cut section cut in it. So we have inner circular smooth muscle fiber and outer longitudinal. In between, we have nerve fiber and nerve cells, minor nerve plexus, which is called Erbach plexus of nerve. Now we have reached to the end of the esophagus. Esophagus is formed of 4 layers. Let's remember what is characteristic for each layers. As regarding the mucosa, we have epithelium. Epithelium was stratified, squamous, non-keratinized. As regarding lamina propria, we was having mucus acini, which is called the cardiac gland, which was present in the upper third and lower third. As regarding submucosa, we was having mucus gland, which is called esophageal gland. It is in the whole circumference. What about muscularis externa? Muscularis externa will be completely smooth in the lower third of the esophagus. It will be formed of inner circular and outer longitudinal smooth muscle fiber. But here in the upper third, it is swallowing process which is voluntary. So it will be formed of a skeletal muscle. The middle third will be formed of mixed skeletal and smooth muscle fiber, while the lower third will be smooth muscle fiber purely. Finally, esophagus is not covered by peritoneum, so the last layer will be adventitia, except the lower 2 cm, which will be covered. The lower 2/3 will be covered by serosa. What is serosa? It will be connective tissue covering by simple squamous epithelium, which will be called mesothelium. Okay? This was the end of the esophagus structure, histological structure. Do we have a medical application for the esophagus? Yes. Medical application, we have a medical application called GERD. I think many of us complain from this GERD. What is GERD? It is gastroesophageal reflux. What is mean by gastroesophageal reflux? It means that the lower sphincter of the esophagus is incompetent. It may make reflux of the gastric acidity onto the esophagus, which make a heartburn. Why heartburn? Because the epithelium of the esophagus is not accommodated to this HCl or gastric acidity. So it will lead to erosion of gastric mucosa. So it gives a sense of heartburn. So what is the medical application? It is a GERD, which is gastroesophageal reflux due to incompetence of the lower esophageal sphincter. If this case is recurrent very frequently and don't undergo treatment, it will cause the epithelium of the esophagus to change into other type of epithelium. This is a metaplasia, changing of epithelium to another mature type of epithelium. And this is a case is called Barrett esophagus, which is a precancerous lesion. Let's summarize or say what is key fact you must remember about the esophagus. It means that esophagus is lined by stratified squamous epithelium non-keratinized. Submucosa contain mucus gland. Muscle is striated, which means skeletal in the upper third, mixed in the middle third, and the lower third is purely smooth muscle fiber. Lower esophagus is an important common site of disease, particularly ulceration, stricture, and cancer, which is due to GERD, which is gastroesophageal reflux disease due to incompetent lower esophageal sphincter. Now let's talk about the second part of the. Now we will talk about the second part of our GIT tube or gastrointestinal tract. It will be the stomach. It is the dilated part To talk about the stomach, we must know the function of the stomach. Stomach is the dilated part of the gastrointestinal tube. What is the function of the stomach? You know, the main function of the stomach is to secrete HCl. And also, not only HCl, it also will secrete digestive enzymes. So it has two functions. First one is secretion of HCl. The second one is mixing with digestive enzyme. And in order to make the food mix with this, it will have in order to mix this food to make a chyme. What is chyme? It is very soft digestion of our swallowed food. So it must have a muscle in order to mix this food. Let's now talk about the stomach and what is the histological structure of the stomach. The stomach anatomically will be divided into four parts. The first part, which is the entrance from the esophagus to entrance of the esophagus to the stomach. This part will be cardia. This part, which is elevation beside the esophagus, is called the fundus. And finally, we have the main part, which is the body. This body ends in another part, which is pylorus, which is the ending of the stomach. Histologically, fundus and body have the same structure. Both are similar, so both of them are called fundus, while pylorus and cardia is having the same structure. So we are talking about the same structure and call them pylorus. So histologically, stomach is divided as a histology into a fundus and a pylorus. Let's see the stomach by naked eye. What is the main structure when you see the stomach in a cadaver or an animal? What is the naked eye appearance of it? Hmm? This is what we see by naked eye when you are looking at the stomach. By naked eye, you are seeing elevation. This elevation is called rouge. So, stomach by naked eye showing elevation. This elevation is called rouge. By scanning electron microscope, when we are going to see this wall, we are going to see an opening inside this wall. This opening is called gastric pit. So, stomach by naked eye is showing rouge. By naked eye, it is showing an opening, which is called gastric pit. Okay. So when we are taking a specimen under the microscope, we will see that there is elevation which is causing the rouge. This elevation is formed of the mucosa and submucosa. To talk about the mucosa, we are going to see that the mucosa of the stomach is mainly secretion, So all the epithelium lining of the stomach will be simple columnar epithelium. Okay, let's go and apply our scheme on the stomach, which is a dilated tube. To talk about the stomach, we are going to divide it into two parts, fundus and pylorus. Let's start with the fundus and the body of the stomach are both of them having the same structure. Both of them is called the fundus. So what are the layer of the fundus of the stomach or the wall of the fundus of the stomach? It will be formed of four layers. What are the four layers? They are mucosa, submucosa, muscularis externa, and serosa, as it is covered by peritoneum covering. Let's talk about the mucosa. What do you know about the mucosa? Yes, it is formed of three layers. Okay, what are they? They are epithelium, which here is simple columnar epithelium because it is secretory. Secrete what? Secrete HCl and digestive enzyme. After that, we are going to have connective tissue, loose connective tissue, containing blood vessel, nerve, and lymphatic, which is called lamina propria. Lamina propria here containing an important structure which is called fundic gland. The third layer of the mucosa is the muscularis mucosa, which is formed of inner circular and outer longitudinal smooth muscle fiber. After mucosa, we have submucosa. Submucosa is formed of dense connective tissue with marginal plexus of nerve fiber. Okay, the third layer is the muscularis externa, and as you know, it is formed of inner circular and outer longitudinal. In between, we have a mesenteric plexus, which is called Ehrbach plexus. But here, the stomach needs another layer of smooth muscle fiber. Why? In order to mix the swallowed food with the digestive enzyme and HCl. So inner circular and outer longitudinal smooth muscle fiber is not enough. We are going to add another muscle fiber, which is inner oblique layer, in order to help the stomach to digest food in all directions. So, muscularis externa of the stomach is characterized by having three layers. It is an important piece of information. This three layer is inner oblique, middle circular, and outer longitudinal smooth muscle fiber, in order to help mixing of the food with the digestive enzyme and chyme. Finally, we have serosa, which is loose area connective tissue with simple squamous epithelium. Now let's see a diagram for mucosa and submucosa. We say that mucosa is formed of three layers. What are they? They are epithelium, which is simple columnar epithelium. We are given here simple columnar epithelium lying on connective tissue called lamina propria, and we have muscularis mucosa. These are the three layers of the mucosa. Okay, can you see here that epithelium was simple columnar epithelium start to make invagination, and they become branched to go beneath in the tips of the lamina propria. So it make a gland, which is deep invagination of the epithelium. This gland is just like a tube, branched tube. So this gland is called fundic gland. It is present in the fundus. Can you see that this fundic gland is a simple tubular branched gland and opening onto the surface by an opening. This opening is called gastric pit. Can you remember in the scanning electron microscope when you see an opening? This is the opening, the gastric pit. What makes the gastric pit? The invagination of the simple columnar epithelium of the stomach into the depths of lamina propria in order to form fundic gland. Fundic gland is a simple tubular branched gland occupying the main depths of lamina propria. Can you see here that the pit, the opening of the gland is short? It is short, straight, and narrow, and perpendicular to the surface. It is short, straight, and narrow, perpendicular to the surface. Okay, this pit, this an opening. It is short, straight, and perpendicular to the surface. It nearly occupies small part of the gland. If this is the gland, it occupies nearly quarter of the gland. So the gland here can be divided into three parts. What are the parts? This is the opening, which is very narrow. So it is the isthmus. Okay, let's remove this. Here we are going to divide the gland into three parts. The first narrow part, which is called the isthmus. And here is the entrance. It is the neck. And finally, we have the remaining part, or the lower part, which is the base. So here in this photo, we are going to have a fundic gland. What is fundic gland? Invagination of the epithelium, which become tubular and branched, opening to the surface by a gastric pit. This pit, which is the opening, is short, narrow, and perpendicular to the surface, occupying quarter of the gland. So this is the fundic gland. What is the parts of the fundic gland? They are the isthmus, neck, and base. After that, after mucosa, there is submucosa, which is loose connective tissue. And then we have musculosa or muscularis externa, which is formed of 3 layers: inner oblique, middle circular, and outer longitudinal muscle fiber. Here, this is under the microscope. What can we see? We can see cells, many cells. This part, it is highly cellular. This is the stomach. This is the fundus of the stomach. How we know? We have here simple columnar epithelium, this part which go and make invagination to be formed of epithelium and go to open by a bit. This bit is short and narrow and perpendicular to the surface. It nearly occupy quarter of the whole length. This is, okay, let's repeat it again. Here, this is the gland which may make invagination and go to make a tube. Here is the gland. Can you see? The gland opened by an opening. This opening is short, narrow, and perpendicular to the surface. And we have a branched tubular gland. So this is the fundus. More magnification to see that this is simple columnar epithelium going making invagination, row of cells, and having a short, narrow pit opening onto the surface. This is the cells lining the gland. It is tubular gland. It is straight. It is like a chain beside each other. This is another photo for the fundus gland. The fundus gland is a tubular, simple tubular branched gland which open by a bit which are short and narrow. The gland are straight and perpendicular to the surface. What are the part of the gland? As we mentioned before, they are isthmus, neck, and base of the gland. Here, the simple columnar epithelium is going to be divided into different type of cells according to their function. Can you give me 2 examples of cells to know? We are going to have a cell which secrete HCl and another one which secrete digestive enzyme. Let's see this type of cells. We are going to have different type of cells to form what is called gastric juice. What is gastric juice? It is a combined secretion of mucus, HCl, and digestive enzyme. HCl here will be secreted from a cell called parietal cells. So we have a cell which secrete HCl. This cell is called parietal cells. We have another cell which secretes digestive enzyme. It is the sheath cell. In order to protect the wall of the stomach from digestion by this digestive enzyme and HCl, we have a cell which secretes mucus for protection. So we have a gastric juice formed of mucus, secretion of mucus cell, parietal cell, and sheath cell. The simple columnar epithelium of the stomach will be lined by 6 types of cells. According to their function, we are going to start with the first type and important one of them, which is the surface mucus cell. From its name, this cell is going to secrete mucus. Any cell which secretes mucus, it will have basophilic cytoplasm. So surface mucus cell, is it columnar, triangular, or rounded cell? It is a part of simple columnar epithelium. So it will be formed of simple columnar epithelium, which will secrete lubricated and protective mucus. Okay, what is the shape? It is a columnar cell which having basophilic cytoplasm and secrete mucus which is protein in nature. It is a mucus which is a glycoprotein, so it will be BAS positive. which will be stored as secretory granules, apical secretory granules, which is BAS positive. By EAM, by electron microscopy, you will find that there is a basal rough endoplasmic reticular mitochondria supranuclear Golgi apparatus. In the apical part, you will find the secretory granules, which is mucin, and it will be electrolucent. All the cells of the stomach will be attached together by tight junction, which is zonular occludence. Why? In order to prevent escaping of gastric juice into the wall of the stomach and digest itself. Okay, so the first type of the mucus, it will be called surface mucus cell. Can you guess where to find it? It will be on the surface, on the first two parts of the gland, fundus gland, which is the pit and the isthmus. By light microscope, it will be a columnar cell containing secretory granules. Here, this is the surface mucus next cell. We have another type of mucus cell. It will be according to its side. It will be in the neck of the fundus. This is called the mucus neck cell. From its name, where to find, it will be found in the neck. It has the same description as its sister, which is the mucus surface cell, except that here the nucleus is flat and basal. They secrete mucus, which is acidic. Take care, here the mucus is acidic for lubrication and protection. So here we have a mucus neck cell. Where to find? It will be found in the neck of the fundus gland. It is the same as a mucus surface cell, except that the nucleus is flat and basal. What about the type of mucus secreted? The mucus secreted here is acidic. Let's see a photo. Here is this a photo of EM. Can you see here that the nucleus is flat and basal? It secretes a mucus. So in the mucus neck cell, you can write that it has a flat and basal nucleus. It has secrete mucus, which is acidic in nature. Now let's go to a medical application related to the mucus surface and the mucus neck cell. What happens if you have a drug or drug or what something that may cause overstimulation of HCl and cause damage to mucus surface cell, which can't produce mucus. So HCl and digestive enzyme will cause painful erosion of the gastric and duodenal mucosa. This may be due to lower secretion of mucus by mucus surface or mucus neck cell or overproduction of by HCl. This is a case called peptic ulcer. What is peptic ulcer? It is a state of imbalance between mucus production and HCl production. If there is decrease of mucus or increase in HCl, it will lead to erosion of the gastric mucosa, which will be very painful and will lead to a clinical case which is called peptic ulcer. The third type of cell, and also it is important, it is the stem cell. What is important to know about the stem cell? Stem cell, as you know, it is responsible for regeneration. Important to know about the stem cell, the position or the site of the stem cell, where to find it in the fundus gland. You are going to find this stem cell in the neck of the fundus gland, which will go division to go upward and downward in order to make complete regeneration of the epithelial cell of the fundus gland in nearly 2 to 6 days. So what is the structure of any stem cells? It is a part of simple columnar epithelium. So it is a columnar cell having oval active basal active nucleus. It has basophilic cytoplasm. Why it is basophilic cytoplasm? Due to the presence of free ribosome as it is a stem cell. Does it make any secretion? No, it hasn't any secretory granules. It is a stem cell. Its function is mainly regeneration. Don't secrete any secretion. Okay, let's go to the second type of cell. We are going to talk about another type of cell, which is a cell responsible for secretion of hormone. A cell which secretes hormone into the circulation, it is called enteroendocrine cell. It is in between enteric epithelium, so it is enteroendocrine cells. It is low columnar cell of two types. If it is lying in between the cell, sometimes it may reach to the surface, so it is open time, which reaches the lumen of the stomach. The second one, which is a short cell embedded in between the cell and can't reach to the surface, so it is a closed type. This cell is going to make its secretion into the blood or into the lumen. It is endocrine, so it will make its secretion into the blood. So here you will find basal granules. Why it is basal, not apical? Because it will release its secretion into lamina propria. Why lamina propria? Because it contains blood vessel. So this cell is very characteristic. Why? Because it has basal secretory granules in order to release its hormone into the circulation. Is there any type according to the type of secretion? Yes, there are many types. The most important is an enteroendocrine cell called G cell, which secretes gastrin hormone. Another one, D cell, which secretes somatostatin. Another one is A cell, which secretes glucagon hormone. Enterocolic cells, EC cells, which secrete serotonin hormone. What about the histological structure? Histological structure means light microscopy and electron microscopy. By light microscopy, it is a low columnar or triangular cell. It having a clear cytoplasm because it has different embryological origin from other cells. It has a rounded nucleus. And don't forget that it has a basal secretory granules. It has a characteristic important stain to know. It can be stained by silver stain due to presence of its basal secretory granule. These granules like silver stain, so it is argyrophilic granules. By electron microscopy, we are going to find that it is containing RAR mitochondria and not supranuclear. It is infranuclear Golgi. Why? Because it will make its secretion basal, not apical. If it will secrete apical, it will be supranuclear. But here, it will secrete its secretion basal, so it has an infranuclear Golgi complex, and it is important for MCQ. Okay, what is the function of this enteroendocrine cell? This will secrete different type of hormones that into the blood vessel of the lamina propria to affect the nearby cells. Like gastrin. Gastrin will stimulate parietal cell to secrete HCl and stimulate gut motility and regeneration. All these hormones we are going to discuss clearly and then action in the physiology section. Here we are going to have a section of the funded gland. Can you see that these cells, triangular cells, taking the silver stain? What is the cells in the funded gland which takes the silver stain? It is the enteroendocrine cells. So enteroendocrine cells can be clearly demonstrated by silver stain cells. It is a triangular cell, sometimes reaches the apex, which is open lumen, which is open type, sometimes don't reach the lumen in the closed type. Let us see the electron microscopic picture of the cell. Can you memorize what can we see? We are going to see electron dense granules, secretory granules. Where to find these secretory granules? Is it apical? No, you are going to find this secretory granule basal because it will be secreted here in the blood vessel. So this is basal secretory vessel characteristic for the electron microscopic photo of enteroendocrine cell. Let's move to see another cell which is one of the most important cell of the stomach. Now we are talking about an important cells which is important in production of gastric part of gastric juice, which is HCl or hydrochloric acid. This cell is called parietal or eccentric cell. Parietal or eccentric cells, why it is called eccentric cell? Because it like the color acidophilic. So it is acidophilic cell. This is the only acidophilic cell in all the cell of the stomach. It is abundant in the fundus gland, but you can find it more numerous in the isthmus and the neck of the fundus gland. By light microscopic picture, you will find this cell triangular in shape. Can you see here this cell? It is highly acidic. It is highly red. So this cell is triangular round cell with acidophilic cytoplasm, and as we see here, the nucleus is rounded central and vesicular nucleus. By electron microscopy, this cell, as it is going to produce HCl hydrochloric acid, it needs more energy. So it has abundant or numerous mitochondria with a small amount of RAR and Golgi complex. It has three important characteristic features, which is some system of tubule and vesicle, microvilli in the intracellular canalicular system inside the cell itself. It has actin filament. Okay, why all of this? Why there is tubular and vesicular system? There is microvilli in the canalicular system. This is for increasing the surface area. Let's see. This is the cell. In order to increase the surface, we are going to have this triangular system. So the cell membrane is going to make a canal. It is a small canaliculi. This is called canaliculi inside the cell and in between the cell. So inside the cell is called intracellular. In between the cell is called intercellular. We are going to have vesicular system, small vesicle. Why this is tubulovesicular system? In order to provide spaces to increase the surface area for production of HCl. In order to prevent this produced HCl from escaping in between the cell, there is a junction complex. What is a junction complex? It means that it is formed of zonula occludens, zonula adherens, and desmosomes. Let's see more picture for the parietal cell. Here we can see that we're having a triangular cell full of many mitochondria. Is it going with parietal cell? Yes, it has numerous mitochondria. It's having central vesicular nucleus, having canaliculi, a channel inside the cell with a small vesicle surround. Why? In order to communicate with the channel to increase the surface area. Can you see here? This is the channel. It has a small microvilli and have tubulovesicular part. Okay, let's see the function of this tubulo. Now, let's talk about the canaliculi and tubulovesicular system. What are their function? Their function is to increase the surface area for production of HCl. So, if we are going to have two types of cell, resting and active parietal cell, can you guess which type is going to have many tubulovesicular and canalicular system? Yes, in the active cell, can you see here that we are going to have many branched tubulovesicular system and canaliculi? Why? In order to increase the surface area. But if I am resting, I don't need. So I will not make this canaliculi or tubulovesicular system. So I have less canaliculi or less microvilli. I'm going to have actin filament. Why? Because actin filament will help me to keep the shape, my shape of canaliculi and tubulovesicular system, and also help changes between microvilli and tubulovesicular system. Let's see another picture for resting and active parietal. Here, can you see? This is a parietal cell, and it is divided into two halves. Some part is resting, one half is resting, and the other is active. Can you guess which structure we use to guess that it is active or resting? Yes, it is the intracell canalicular system and tubulovesicular system. If there is extensive and branching canalicular and tubulovesicular system, so it is active. If there is less canalicular and intervesicular system, so it is going to be less. So let's see if your answer is right or wrong. This part, the left part, is the resting because it has non-use or many few microvilli and many physical system. But in the second part, it is active because we can see many vesicle, but we can see a highly branching canalicular system and many microvilli. Now, what is the function of the parietal cell? The main function we need is the hydrochloric acid. HCl play an important role in protein digestion and lowering pH in order to activate the digestive enzyme and kill bacteria and virus. This is produced by the mitochondria, canalicular, and tubulovesicular system. Does it has another function? Yes, it produces an intrinsic factor, which is a protein in nature. This intrinsic factor binds to vitamin B12 and helping its absorption in the small intestine. So, what organelle do we need for production of this intrinsic factor? It is a protein in nature. Excellent. We need RAR and Golgi complex. In addition to the mitochondria, which is present numerous in the parietal cell for production of HCl. Now, let's go to the medical application of parietal. What is this medical application? We say that the function of parietal cell is production of HCl and intrinsic factor. If there is damage to this parietal cell, it will lead to decrease in secretion of not only HCl but also for the intrinsic factor. So our body will not be able to absorb vitamin B12 and lead to a type of anemia which is vitamin B12 deficiency and will lead to megaloblastic anemia. This was the parietal cell as it produces HCl. There is a main component of the gastric juice which is the digestive enzyme which is produced by a cell called sheath or peptic cell. Now we are going to talk about the sheath or peptic cell which is responsible for production of digestive enzyme which is pepsinogen enzyme. This cell is a typical protein secretory cells. Okay, what are the character of a typical protein secretory cell? It is a columnar cell having basophilic cytoplasm, having rounded basal nucleus, they're having apical acidophilic zymogen granule. This is the character of any protein secreting cells, which by electron microscopy you are going to find RAR mitochondria supranuclear Golgi apparatus. So, chief cell or peptic cell is a typical protein secreting cells. It is pyramidal basophilic cytoplasm, apical acidophilic zymogen granules. By EM, there is RAR mitochondria Golgi complex. This cell, as any simple columnar cell of the stomach, is attached to the surrounding cell by the occluding junction or tight junction. Okay. Where to find this cell inside the funding gland? Is it common like parietal cell in all parts? No, it is just present in the base of the gland, in the base of the funding gland. So in the base of the funding gland, what type of cell can you find? It is the sheath or peptic cell. This cell produces a protein. It is a typical protein secretory cell. What's protein? It is the pepsinogen protein. It is in an inactive form of digestive enzyme, which will be changed into a pepsin, which is the active enzyme inside the lumen of the stomach in the acidic media on HCl produced by parietal cell. So, chief cell is a typical protein secreting cells. You have to know that it is present inside the funding gland in its base. Its function is mainly secretion of pepsinogen, which is become activated in the lumen of the stomach in the low pH. It also secretes an enzyme which is lipase enzyme. Can we see a photo for the sheath or peptic cells? Yes. It has a basophilic cytoplasm and apical acidophilic zymogen granule. It has active nucleus. Okay, can you see the arrow here is identifying the chief cell or basal cell. Okay, by this chief cell, we came to the final cell of the simple columnar epithelium of the stomach. Let's go to complete the structure of other part of the stomach. We talk about the fundus. We say that it is formed of mucosa, and we talk about the fundus gland by its cell lining or its six type of cell lining. We talk about submucosa. We talk about the muscularis externa, which was showing a third layer of muscle, which is inner oblique muscle, in order to allow mixing of the food. And finally, we have serosa. Now we are going to talk about pylorus, which is formed of three parts. As regarding it is formed of the pyloric itself, pylorus proper, which is the terminal part, then the antrum, and finally the pyloric sphincter. What is the microscopic structure of it? It will be formed of mucosa, submucosa, muscularis externa, and finally serosa. Here, mucosa is like that of the fundus. It is simple columnar mucus secretory epithelium resting on lamina propria. Here, there is no funding gland. There is pyloric gland, and we have to know the difference between funding gland and pyloric gland. We have muscularis mucosa, which is arranged as inner circular and outer longitudinal smooth muscle fiber. Then we have submucosa, which is formed of dense connective tissue with Meissner plexus. Okay. What about muscularis externa? What about the function of muscularis externa? Can you see here that in this photo, what is the main target of pylorus? The main target of pylorus is to constrict, to close the opening. For what? In order to keep the swallowed food for time enough inside the stomach, nearly 3 hours. Why? In order to allow time for the stomach to mix the food with HCl and digestive enzyme. So the main target of the pylorus is to close the opening for the swallowed food to pass into the small intestine. So here, muscularis externa will be formed as usually of inner circular and outer longitudinal. But here, the inner circular will be more thickened. Why? In order to form a pyloric sphincter, in order to close the passage of food till it became chyme. So here we have that the inner circular becomes thickened to form a pyloric sphincter. In between the inner circular and outer longitudinal, we have a meninteric nerve plexus, which is the Erbach plexus. Finally, we have serosa as usual. Okay. Now let's talk about the mucosa. We say that the mucosa is formed of epithelium, lamina propria, and muscularis mucosa, and we see that lamina propria contain the pyloric gland. What is the difference between pyloric gland and fundus gland to see them in? Here is a photo of a pyloric gland. What is a pyloric gland? Is it a simple tubular branch? Can you remember fundus gland? It was like that. Simple columnar epithelium go down and make invagination and form a tube like that. Here, this is not as simple as that. It is simple columnar epithelium go down and not become branched, it become coiled. So it is a branched coiled gland. What about the bit? The bit here, the opening of the gland is large. It is nearly half to half. Can you see here the opening? Half of the gland is opening and half is the gland. So the bit to gland ratio is 1 to 1. Okay, so the gland is coiled and lined by all type of cell except chief cell. Okay, where to find chief cell? Chief cell is present only in funding gland, and this is if you have a comparison between pyloric gland and funding gland, not to miss, or even an MCQ multiple choice question, you have to memorize that no chief cell in pyloric gland. So pyloric gland is a branched coiled tubular gland. which its opening or pit is wide and long. It nearly occupies half of the body of the gland. That says the pit to gland ratio is 1:1. What about the lining epithelium of the gland? It is simple columnar epithelium containing different type of cell like the fundus gland except for chief cell. You see here, this is a fundus gland and this one is pyloric gland. And you see here that the fundus gland is simple tubular gland. It is like a chain beside each other. And the gastric bit is nearly quarter or 1/4 to the body of the gland, and it is narrow, straight, perpendicular to the surface. While the bit of the pyloric gland, it is large one and wide one. It is pale stained. It nearly occupies half of the gland. Okay, let's see it in a section. Can you see here? What is this? This is fundus or pylorus. I will give you a second to think. Here, this is the mucosa of the fundus and pylorus. This part and this part is occupying the gland, which is maybe either fundic or pyloric. How to demonstrate? Fundic gland is a simple branched gland with the pit occupying nearly quarter or 1/4 to the body of the gland. Can you see here that gastric pit occupying in relation to the gland? Is it 1:1 or 1:4? It is 1:4. So this is fundic gland. What about this? It is highly coiled. This is the gastric opening. This is a bit, it is large. Can you see here that as it is coiled, there is many TS section here? Why TS section here? Because it is coiled. So when you cut in the tube, it will appear as rounded section. Moreover, can you see that the basal part of the fundic gland here in this photo appear basophilic? Why? Due to the presence of a cell called chief cell, which is present only in the fundus while it is absent in pylorus. So this photo is fundus, fundic gland, while this photo is pyloric gland. Okay, let's go to know structure or function of the stomach. Memorize the main function of the stomach according to structure, function, adaptation, and it is an important question. You have to memorize what we have in order for the stomach to act as a reservoir. We have a strong pyloric sphincter and we have folds which is called reject, which can help in distension to give more spaces for the food. What will make the stomach act as a mixer for mixing food in the fundus or the body of the stomach? We have a third inner oblique layers in order to mix the food in the three directions. The fundus, muscularis externa of the fundus and body, showing that we have an inner circular, middle, inner oblique, middle circular, and outer longitudinal muscle fiber in order to mix the food with the enzyme and HCl to convert it into chyme. What is for the digestive enzyme? We are going to have a parietal cell which secretes HCl. We're having a chief cell which secretes enzyme pepsinogen, lipase, and renin enzyme. Other function includes secretory function. Does the stomach have a secretory function? Yes, it secretes intrinsic factor. Can you memorize? Yes, intrinsic factor is also secreted by parietal cell. Its deficiency may lead to megaloblastic anemia due to decreased absorption of vitamin B12. Another secretion of the enteroendocrine cell is secrete different hormones like serotonin, endorphin, and somatostatin. And for protection of the stomach from HCl and digestive enzyme gastric juice, we have a protective function. How? Because secretion of HCl will kill bacteria which may be present in the uncooked food. Also, if it secretes HCl, we have a mucus which prevents the damaging of the layers of the stomach. And we are going to have a stem cell in the neck of the glands in order to make continuous renewal of the cell every 2 to 6 days, making continuous renewal of the gland completely. And now we have reached our end of the session. And thank you and goodbye, and seeing you in the next lecture. Thank you.